today's episode of Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Ahlstrom, and we'd like to thank our episode sponsor, Celgene Corporation, for their support of Myeloma Crowd Radio. Now, in the prior show, we gave you a little update on our new product called HealthTree, and our family has been on a 50-city tour. We have now done 44 out of the 50 cities, and most recently in Santa Fe and Portland. And we're happy to share that we now have over 600 patients participating in this HealthTree tool. Now, because everyone's myeloma situation is different and everyone's story is important, HealthTree is a new online tool that provides key benefits to patients, and there are three main benefits. First, it helps you understand your myeloma treatment options that are personally relevant to your situation. Second, if you're unfamiliar with myeloma clinical trials, and those like we're talking about today, it helps you find clinical trials that apply to your situation at that point in time. And we've integrated with the SparkCures tool. And third, it'll allow us as patients and researchers to see patterns of responsive care and help us find ways to live longer with myeloma. So we're so excited to have this tool, and you'll be hearing more about it in the future. We'll be having an intro video come out later in the next week or so, and we're excited to share that with you. It's very important that as patients we become educated on specific therapies, and that's why I'm excited for this show today, because the landscape in myeloma is changing very quickly. We have a lot of new drugs that are coming out into the marketplace and into early clinical trials and mid-clinical trials and even late-stage clinical trials. So the more we know about these studies, the better off we're going to be in terms of picking studies that we want to join. So I'm excited to have Dr. Maria Chaudhry with us today from the Ohio State University and the James Cancer Center. Welcome, Dr. Chaudhry.
Thank you so much, Jenny. Thank you so much for having me here in your show. It's always a great pleasure to talk about new and exciting therapies in multiple myeloma. And I would like to take this opportunity to thank you personally for your wonderful work and the great platform that you have provided for myeloma patients. It's great work. Thank you.
Oh, thank you so much. Well, we'd like to give you a proper introduction before we get started talking about this Glaxo Smith Klein drug. Maria Chaudhry is Assistant Professor in the Division of Hematology at the Ohio State University Comprehensive Cancer Center. She is new to the James Cancer Center, having come most recently from MD Anderson in Houston. She is originally from Pakistan. Her awards include a Georgetown University Hospital Award as Chief Fellow in the Hematology Oncology Fellowship Program. She is a review editor and editorial board member for blood cancers publication called Frontiers of Oncology. She's a member of ASH, ASCO, and the American College of Physicians. She serves on several committees at Ohio State University, including the Clinical Scientific Review Committee. Dr. Chaudhry has been actively working on clinical trials, including new proteasome inhibitors like carfilzomib, myeloma genetics, and new antibodies like the one we will be discussing today. So again, thank you so much for taking time out of your busy schedule. I don't know how you as researchers do it, because you're seeing patients and you're performing research and you're speaking at conferences, and it's just a lot. So we're just really appreciative for all of your dedication around myeloma.
Thank you so much. It's an honor to work in this field and serve the patients.
Well, we're happy that you do. So we are talking about a specific drug today, and this Glaxo Smith Klein drug kind of caught my attention and a lot of people's attention at the most recent ASH last December. And later this year, the FDA granted them this breakthrough therapy designation. And the drug name is called, it doesn't even have a name yet, it's just a number, GSK2857916. So maybe you just want to give us a broad overview about what this drug is and what it does, and then we'll go into other questions after that.
So this medication, this drug is what we call antibody drug conjugate. So there is a difference between antibody drug conjugate, monoclonal antibody, and what we call BiTE antibody. So the difference is that this drug GSK drug is a antibody drug conjugate, and it has a monoclonal antibody part with a payload attached to it, which means that the antibody part of the drug targets a specific protein, a specific receptor on the surface of cancer cells, and deliver the drug or a toxin attached to the antibody close to the cancer cells. So in case of this drug that we are going to talk about today, the GSK monoclonal antibody drug conjugate, the antibody targets CD38. Antibody targets the BCMA, or B-cell maturation antigen, which is universally expressed on all plasma cells. And the toxin attached to it is called MMAF. When this antibody is delivered to the patient, the CD38 part attaches itself to the plasma cells, and it is internalized. And the toxin then kills the plasma cells. And it also stimulates the immune system because of the monoclonal antibody part. That it is different from a monoclonal antibody like daratumumab or elotuzumab. These are the two drugs which are already approved for multiple myeloma patients in relapsed refractory setting, although daratumumab is recently approved in frontline setting. So a monoclonal antibody has actually targets a protein or receptor on the surface of, again, the tumor cells. And when it attaches itself to the tumor cells, it stimulates an immune reaction and kills the tumor cells or the cancer cells by direct immunogenicity, as well as by activating different parts of the immune system. So that's a monoclonal antibody. And the BiTE, which stands for a bispecific T-cell engager. So it's the antibody that attaches two cells and bring these two cells together. For example, in multiple myeloma, the BiTE antibody that is in clinical trials targets two proteins, BCMA as well as CD3. BCMA is present on the surface of plasma cells, and CD3 is present on the surface of T cells. So when this monoclonal antibody is delivered to the patient, it attaches itself to the tumor cells and brings the T cells close to the tumor cells and cause the toxicity and the cell death. So that's the difference between all these three molecules. So in terms of antibody, yeah. Go ahead.
So just to review, so I know as a patient it's easy to get confused because immunotherapy means this whole realm of products. And now that we're seeing all these things come out, so we were familiar with these monoclonal antibodies, and what you're saying is the monoclonal antibodies are going after this certain protein like CD38, like daratumumab does, on the surface of the cells, and your immune system is actually what kills the myeloma cells. And in the BiTE technology that we're not talking about today, you're joining that same protein target with another target that joins those things together, and they kill each other, basically. But this one that you're talking about today, it's like a dump truck, it targets that protein, that BCMA, and you'll go into this later, I hope, and why BCMA, why that target, and basically dumps this toxin on top of it. So it's hard for us to keep it all straight, but we appreciate you kind of breaking it down into different categories, because sometimes people talk about immunotherapy and then we as patients are like, what? We don't understand the different things. So that was a nice description that you gave.
Okay. Immunotherapy, when we say in a general term, it just means that any molecule, any drug that just recruits or stimulates the immune system against a specific disease. But then you're right, it breaks down into different parts and different molecules and different mechanism of action. So it's all very, very interesting. Complicated, but very, very interesting.
Very much so. So now, earlier in the year, or a few months ago, the FDA gave them this Glaxo Smith Klein drug, or GSK drug, this breakthrough designation. Can you kind of explain for patients what that means and why that's important or not important?
So it's very important, actually. So this FDA breakthrough designation is given to a drug that treats a serious or a life-threatening condition, or being studied to treat a serious or life-threatening condition in which there is an unmet need. important to note that when this designation is given, there is already early clinical evidence which points that the drug may demonstrate substantial improvement in terms of controlling the disease or improving the quality of life or other benefits over what is already available or FDA approved. Okay, so the purpose of giving this designation is to facilitate or expedite the development as well as review of the drugs and eventually approval of these medications. Sponsor or the drug company request this from the FDA, and drug companies usually have early phase trial data, phase 1 or phase 2 data, based on which FDA approves, gives this designation status. And then FDA very closely reviews and helps the drug company to develop the drug. For example, in daratumumab, this FDA breakthrough designation status was given in 2013 based on phase 1 clinical data, and the drug is approved as a single agent in 2015 based on excellent clinical data that was available. So the essential purpose is to facilitate and speed up the process and bring the drug, if it is active, available for the patients as soon as possible.
Mm-hmm. And how long typically from the time they get this breakthrough designation? I mean, does it depend on the drug and the clinical trials that have already been in place, or is there kind of a standard timetable once they get the breakthrough designation? I'm just curious about that.
There is no standard timetable. If you look at all the cancer drugs that have been given the FDA breakthrough designation, the most effective drugs had good data earlier, and they were approved within a couple of years from getting this designation. There were some drugs that had good, effective early phase clinical data, but didn't pan out later. So it's important to note that when this designation status is given, it means that the drug may have potential, but still clinical efficacy and clinical benefit still has to be proven.
Interesting. And I saw that it was being used in relapsed and refractory myeloma, or myeloma that's no longer responding to a lot of different drugs that are approved, and some lymphoma. So I know in leukemia, when you were thinking about like the CAR T drugs, and they're going after different targets that leukemia has that myeloma doesn't have, like CD19. Sometimes, you can't just move it over and use it in myeloma because myeloma doesn't really have CD19. So does lymphoma also have BCMA as a target? I was just curious about that.
So this protein or a receptor, which is called B-cell maturation antigen, is universally expressed on all plasma cells, but also in mature B cells, which are actually destined to become the plasma cells. Okay, so the early phase clinical trial or phase 1 clinical trial essentially included all the hematological malignancies that have some degree of BCMA expression. So they did include the lymphoma as well. I have not seen the results from the lymphoma cohort of the patients yet, so I don't know how active it is in lymphoma. But essentially, that's the purpose of the early phase clinical trial, is to study the drug and see if we can find activity there.
Okay. And can you explain the evolution of maybe a little bit of myeloma? Because I know they start with so plasma cells are the really mature cells, correct?
Yes. So plasma cells are the antibody producing.
Yeah. Okay. So can you explain the evolution of how a cell evolves to be a plasma cell, and then where does this B-cell maturation antigen come into play? I don't think I fully understand that.
So B cells are small white cells. They are the part of our immune system and the blood system. So during their development, B cells are exposed to the antigens and become differentiate further. A portion of these cells, a few of those mature B cells, become plasma cells, and plasma cells are the antibody-producing cells, immunoglobulin-producing cells. That's what we see in multiple myeloma. And B-cell maturation
IgG or whatever.
IgG, all these different types of immunoglobulins. So they are a part of our immune system. B cell maturation antigen is expressed on the surface of B cells that are destined to become plasma cells, as well as universally on all the plasma cells. So it's a protein receptor on the surface of the cells, and the function of this is when it binds to its specific molecules, then it gives growth signals to the cells, and it is important for the long-lived plasma cells. So bottom line, it's needed for the growth as well as maturation of plasma cells, as well as for their long life. It binds to few molecules, and one of those is APRIL molecule. I will talk about it later, and there are drugs that are targeting those molecules as well, and gives the downstream signals. And
great.
One of the great targets in multiple myeloma.
Yeah, it seems like it. So what other drugs are targeting BCMA? And I know a lot of the CAR T-cell studies right now are targeting BCMA. Why is BCMA such an attractive target? And then I have a question about the levels of expression of BCMA, because I know some patients in the early days in the CAR T trials, they would go and get tested, and some of the CAR T trials were only accepting them if they had a certain level of BCMA. So I'm curious, when all these drugs come out and they have this BCMA target, does the level of BCMA really matter?
Great questions. So whenever we target a protein on the surface of cells in any tumor, it's great if that specific target is present on the cancer cells only. And essentially, this is because we want to decrease the toxicity and off-target effect, so that the patient can get that specific drug, a specific molecule for a longer period of time and can get the benefit. So BCMA is expressed, as I said, universally on the plasma cells. So that's why it's a great target in multiple myeloma. And it has been targeted by different types of molecules. You already mentioned CAR T cells. So the CAR T cells directed against BCMA there was great data that was published at ASH, very effective therapy. We need long-term data, as well as phase 2 and later line clinical trials to essentially know the future of CAR T cell therapy, but it looks great. Other than the CAR T cell, we're going to talk about a little bit more about the early phase or phase 1 trial data of this antibody drug conjugate from GSK. This drug conjugate is the most developed molecule so far in terms of BCMA targeting molecules. And then there are bispecific T-cell engagers as well, which are called BiTE antibodies. So they are, again, in early phase clinical trial. There is a molecule that targets BCMA on the surface of plasma cells, as well as CD3, which is expressed on the surface of T cells, which are the immune killer cells, brings them together for increased toxicity and increased cell death and increased activity. So these were the bispecific T-cell engagers. So this is go ahead.
And why is this one considered more developed? Is it just further along in clinical trials?
It's just further along in the clinical trials, yeah. Mm-hmm.
Okay. Wonderful. So let's talk about the early results, if you can, about why it was, because the early results came out, I think, last December. Maybe they came out before that, but that's when I heard about it. And maybe you can describe what those results were and why everyone is so excited about it.
Yeah, so you're right. So we discussed this trial at our ASH meeting last year, again at ASCO in June this year in 2018. So this is an early phase clinical trial, phase 1 trial. So I would mention that the purpose of the phase 1 trial is that we have already seen preclinical activity, or means the drug is active in cell lines or animal models, and now we want to study that in human beings. So essentially the patients that are enrolled or eligible for the phase 1 clinical trials are the patients who have relapsed and refractory multiple myeloma, which means that the patients are progressing on the current line of therapy, or they are progressing shortly after finishing the last treatment.
Okay, so this is a phase 1 clinical trial in which the purpose is to see if the drug is safe in a cohort of patients. One of the endpoints of the study, or one of the goals of the study, is again to look at the responses as well. But the primary is always to look at the safety profile of the drug, to look at the toxicities or side effects that patients will get. So this was a phase 1 trial, and sponsored study in which this medication was given intravenously every 3 weeks, and it was a 1-hour infusion. Looking at the patient population in this clinical trial, so patients had received multiple lines of therapy before. More than half of the patients, about 60% of patients, have received 5 lines of therapy. And about 40% of patients had received daratumumab, and they have progressed on that. Most of the patients
40%, you said?
40% had received daratumumab, yes.
Okay. Wow.
Most of the patients have also received immunomodulating drugs, which are Revlimid or pomalidomide, as well as proteasome inhibitors, which are Velcade and carfilzomib. So this was the patient population which was very relapsed and refractory. So looking at the results of this trial with this medication, the response rates were impressive. Overall response rate was about 60%, which is great in this population.
Yeah, that is great.
And half of these patients had very good partial response, which means that they had more than 90% reduction of their myeloma proteins or better. So that was great as well.
Yeah.
And 40% of patients who have progressed on DARA before have responded in this trial.
Wow.
And progression-free survival, which means the amount of disease control that we got, was about 7.5 months. And then there is another endpoint that we looked at was duration of response, which means when the patients were on the drug, for how long did they get the clinical benefit. So at the time the study was presented, which was about 6 to 7 months of follow-up, a duration of response was not reached, which means that the patients who were responding were doing well and continues to have clinical benefit.
Right. They hadn't relapsed yet, so that's great.
They have not relapsed yet, yeah. They were still on the drug.
And how long was that study?
Study is still ongoing, so they will keep updating us the results. But when the results were presented, median follow-up was about 6 to 7 months.
That's great. Well, that's terrific.
That was terrific, actually, yeah.
Yeah, yeah. That's very exciting as a new option for patients who are relapsing through the myeloma therapies, and especially through daratumumab, because I know patients can get really frustrated when they relapse through that.
That is true, and as we know that myeloma is an incurable disease, so most of the patients at some point relapse, and once patients have exhausted or received proteasome inhibitors, immunomodulating agents, and daratumumab, their prognosis tend to be dismal. So we need better therapies with different mechanism of action. So that's why such good responses and a new drug with a different mechanism of action is very important.
And I think what makes those results so impressive is that it happened as a standalone drug. You weren't giving in the clinical trial. Yeah, you weren't using any other drugs with it. You were just using it by itself.
As a single agent,
right?
Yes, you're right.
Yeah, that's amazing. So the trial that you currently have open, and you have it open at the James, correct?
Yes.
I guess how many trials have this open, and then can you explain more about the trial that you currently have open using this drug right now, and all the kind of the details behind the study, the current study, because that was the phase 1 study, right?
Yes, that was the phase 1 study. Yeah. So now this drug is moving towards the phase 2 part. And this study is actually going to be a multinational study, and it is open at multiple institutes within the America as well as some international areas like in Europe as well. So phase 2 study and patients who have, of course, multiple myeloma but relapsed and refractory disease, which means that the patients who will be eligible for this trial will be the ones who have failed at least 3 prior lines of anti-myeloma regimens, which should include anti-CD38 antibody, which will be daratumumab or any other drug in the clinical trials that target CD38 alone or in combination. And the patients who are refractory or who have progressed on an immunomodulatory drug, which will be pomalidomide or lenalidomide, as well as refractory to a proteasome inhibitor, which will be Velcade, ixazomib, or carfilzomib. So essentially, the patients who have progressed on at least 3 prior lines of anti-myeloma therapy. Patients who have good performance status, which means that they're able to, because it's an early phase clinical trial, so patients who are able to get out of the bed and take care of themselves. So good activity status is important as well. So these are the patients who are eligible for this trial. And then another thing that I will mention, that in the phase 1 study, there was a reporting of side effects, ocular toxicity or eye side effects. A percentage of patients did get blurry eyes, dry visions, and increased sensitivity to light. That was one of the most common side effects that was observed in phase 1 clinical trial. So in phase 2 clinical trial, when we will be treating the patients, we will have our eye department or ophthalmology department work with us. Patients will be getting eye drops as well as eye exams when they come in for the infusions. This is a very important trial. It's a good trial for the patients who have progressed on daratumumab, because this is a patient population who do not have many treatment options available, and the prognosis tend to be dismal. So different drugs with different mechanism of actions are needed. And as I said, that this trial is open at multiple centers. It's very easy to find the centers by going to clinicaltrials.gov. At Ohio State, we have just recently opened this trial as well.
How many sites are available in the United States? You said multinational, and I'm assuming there will be a lot of different sites open, but do you know how many?
I think it's already open in about 4 to 5. I do know that it's open in New York. It's open in Atlanta and Indiana, I think. Yeah, other places on top of my head, I cannot think, but I think that it's in a process of opening at a few other cancer centers as well.
Yeah, that's okay. And we suggest that patients, instead of clinicaltrials.gov, they use a tool called Spark Cures, because it's much easier to navigate. And I know that study is in there, and it will show all of the treatment sites or clinical study sites. But the James is obviously one that it's available at. So when you join the study, because sometimes I think people need to consider traveling for these types of studies and can travel. So what are the travel requirements? How it's an infusion every 3 weeks, and how long are you on those? How long do you get the infusions for?
So great question. So yeah, infusion is every 3 weeks. Infusion lasts for about an hour. But after the patient finishes the infusion, patient will be just observed for a couple of hours more, about 3 hours, and then we let the patient go. The requirement is that when the patient is eligible for the study and the patient agrees to get enrolled for the studies, they will have some screening studies that we can consolidate and do together in few days. And after that, they will be travel every 3 weeks. Another part of the study is the eye exam. Because of the ocular toxicity or eye side effects that the patients had with the phase 1 clinical trial, In phase 2 part of the study, sponsor prefers to get the eye exam as well. So the patients will be scheduled in the eye clinic the same day they come in for the infusion for the eye exam. So the logistics of all these things together varies on patient as well, like varies by patient. But in general, the infusion is every 3 weeks, so it's not that frequent.
And then how many months do you provide treatment on the study?
So essentially, the treatment continues as long as patient tolerates, which means that the patient is not getting any toxicity that will prevent us from treating the patient, or when the patient progresses.
Okay, that makes sense. And I know there are these long waiting lists for these CAR T studies, but in my mind, studies like this may be a great option for patients because it had such a powerful effect. It may be a great option for patients. So I don't know. I would seriously consider that if I were in that situation.
I agree with you. Yeah, and it's a phase 2 study, so when the spots are available, patients can just be enrolled, which is a bit different from phase 1, in which we enroll a certain number of patients and then wait, follow them, make sure that they don't have any side effects that prevent us from continuing further. So it goes by cohort. But in phase 2 study, the spots are open, and the patients, if they're eligible and if they agree, they can be enrolled right away. And you are right, for CAR T cells, it's a little bit different because patients have to be seen and then cells have to be prepared and then infused.
Right, so this is an off-the-shelf approach.
This is, more like that,
So I know in this phase 2 study, you're looking at patients that have had these prior lines of therapy, including, that they've relapsed after getting like Revlimid or Pomalyst or Velcade or carfilzomib and daratumumab, those types of drugs. So it doesn't make sense when you're testing it in this phase 2 study to combine it with anything else. But is there any indication if you could use it earlier, before patients have relapsed on those other types of drugs, what it might pair with well, I guess? Like what other,
that's a great question,
if you were to use it in combination, what other types of combinations could it be considered in?
That's a great question. And actually, we have already started thinking about that, and sponsor has already started the work to combine this drug with other anti-myeloma therapies. So as we know, when the patients with myeloma relapse or when they progress, the disease tends to become very diverse, very genetically unstable, and hard to control. So using the combination of drugs targets the disease from different mechanism of actions and from different angles. And we have seen in past that it controls the disease better. So it's a great idea to combine an effective drug with the other myeloma medications that are already approved. Combinations have to be rational and safe at the same time. So this medication can be combined with proteasome inhibitors like Velcade. It makes sense because of the mechanism of action, as well as it should be studied with lenalidomide and dexamethasone as well. And actually, there is a trial that the sponsor is going to bring forward. It's going to be a phase 1 trial, but the medication, this GSK medication or BCMA antibody drug conjugate, will be combined with lenalidomide and dexamethasone or with Velcade and dexamethasone. I think it's a great idea to combine with this drug with daratumumab, which is an anti-CD38 monoclonal antibody, to target both the BCMA antigen with this drug as well as CD38 receptor with the daratumumab. And
that makes sense. Going after 2 different protein targets instead of just one.
Yes, and then there will be another trial that will combine this anti-BCMA antibody with the PD-1 inhibitor. So as we already know, the T-cell exhaustion or immune cell exhaustion happens in cancers, especially in multiple myeloma, when patients' immunity weakens and they cannot build an immune response against the cancer cells. So PD-1 inhibitors are actually release the brakes on these T cells, and they can target and recognize the cancer cells. So it makes sense to combine the monoclonal antibodies with the PD-1 inhibitors as well. So
well, you have a lot of experience with PD-1 and PD-L1 inhibitors at the James, so that would be a great study for you all to run, I'm sure.
That would be great as well, yeah. So there are different things that we are looking at, combining these medications with other anti-myeloma therapies. And this is all very promising, and I think this drug will transform the treatment paradigm for multiple myeloma patients. And I hope it will, and hopefully will improve the disease-free survival for our patients as well.
Oh, I know there's so many things like this coming out that are so exciting. So I think there's a lot of hope for myeloma patients. I just have a few follow-up questions for the phase 1 study. When I know they're looking at safety when they're testing in phase 1, and you're also looking for some indication, and they had these great results come out. Do they ever go back and look at the genetics, like for patients who had the better responses, what sort of myeloma genetics they had, so they can see if it's better for a specific type of patient? I know by the time patients have relapsed multiple lines of therapy like that, the genetics of their disease are pretty complicated, and they might have more mutations than they had before, or different combinations of mutations. But was there any insight there about the genetics of myeloma that you know of?
So that's a great question. With the phase 1 study, nothing has panned out. But with the phase 2 study, we will be collecting the patient's samples to look for genetic mutations and correlate those with the responses, because it will be great to know that which patients respond better than others. So it would be great to know. But we don't have any information from phase 1 study yet. And I will also add, I think you asked me about BCMA expression as well. So in the phase 1 study, the patients were enrolled irrespective of their B-cell maturation antigen expression on their tumor cells, because we have seen activity in patients or in tumors that have low BCMA expression, because the binding of this antibody drug conjugate with the myeloma cells are very strong. So even if there is low expression, it still works. So for phase 2 study, there is no cutoff or limit for BCMA expression.
And what does that mean anyway, the BCMA expression level? I guess I don't understand the relevance, because, it seemed to be this big deal when the CAR T stuff was first introduced maybe a year and a half or 2 years ago. And now it seems to be like, well, it's going to work regardless of the level of expression. So I guess, can you help answer that question? What does that mean, the level of expression anyway? And is it important at all? It doesn't sound like it is.
So level of expression means using specific testing, staining on the cancer cells. We look at the samples and see how bright the expression is. And the pathologist and oncologists give it a number, like there is 10% expression or 50% expression, which means that this is the percentage of cells that express that particular antigen, most probably, or this is how bright the staining was with this, for this specific antigen or for this specific protein on the cancer cells. Because the thought always is that if there is a weak expression, if there is weak staining, is the drug going to work or not? Okay?
Right.
So because if the staining is weak, then do you need more like high doses of the drug versus low doses of drug, or it's not going to work at all? So that's what the concept is. So when the drug was started, at the baseline, sponsor as well as the investigators were looking at the BCMA expression and seeing if they can correlate this with the responses. but didn't pan out that way. Does it make sense?
So at the beginning of the phase 1, did it? Yes, it does completely.
Mm-hmm.
But yeah, because if there's a lot, will it work? Or if there's little, is there anything for it to go after anyway? But so do you test in the phase? They tested in the phase 1 and phase 2. Also, the study you're currently running, they measure everybody's BCMA levels. And was there any indication in the outcome data on the phase 1 if they measured the BCMA levels, like patients with really heavy BCMA expression seemed to respond better, or they were more durable responses, or was there any relationship there that they saw, or is there any relationship that you're seeing in the phase 2?
So for the phase 1 study, when they presented, they have not reported in terms of the BCMA expression the results that I remember. But they did mention that the degree of response had no association with the BCMA expression. Again, the reason being the antibody binds with the receptors tightly, irrespective of the degree of expression. So the drug still works.
Yeah. Okay, great. And you talked about this at the very beginning, that it delivers this toxic payload of MMAF. Do you want to explain what that is? And it looks like, I don't know, I've always heard about, MAF in myeloma, so it's not related, right? That you talk about the MAF translocations in myeloma, that's not related to this type of toxin. It just happens to have a similar name?
No, it's not related to that. So this toxin itself cannot penetrate the cancer cell, cannot penetrate the myeloma cell. Okay, and the mechanism of action is that it's an anti-tubulin agent, which means that it inhibits the cell division and cell synthesis. So this antibody delivers that toxin, and then it's internalized. It's taken up because it binds to BCMA. It is taken up inside the cells, and that's how the toxin gets inside the cell.
Well, that's nice because it's specific delivery, and that's what we want.
It's specific delivery into the cells, yeah. It's a very, very smart concept, yeah.
Yeah, well, we love it, and we're looking forward to it, and this sounds like a really fabulous study and one that patients should really consider joining. Is there anything else that you want to share about this study in particular in terms of how patients get into the study, or anything else patients would need to know about joining the study?
I think if the patient is coming from a long distance, or if they have to travel in order to get to the cancer center who has the study, then it's important for them to know if they will be eligible based on what they have received before and other factors. So I think Spark Cures, as you already mentioned, is a good resource for that. We have this trial here at the James Cancer Center. So I'm happy if anyone wants to contact me directly. I can give my email. My email is my first name, maria.chaudhry@osumc.edu. Patients can approach me as well. And then go from there. It's very important to enroll in the clinical trials. I always encourage at any stage in their disease, I always encourage patients to enroll in the clinical trials. This way we can get better drugs approved, but also the patients can get drugs on the clinical trial that will be later approved and will be available in the community. And they can get drugs earlier while they're on the trial. So I always encourage that.
Well, we're huge advocates, and that's actually why we started this radio program, because I think 3 to 5% of myeloma patients join clinical trials. And now we have these great drugs like daratumumab and these CAR T-cell drugs and this GSK drug coming through clinical trials. And the faster that you can have patients participate, the faster we can come to conclusions about what's going to be potentially curative for us. So I think I agree that at every stage, if you are newly diagnosed or if you're just your first relapse or a multiple relapse, I mean, there's really a lot of different studies that you could consider. And Spark Cures does have a concierge kind of service where you can call a phone number also if you want to join a clinical trial. But you're very generous to give out your email, and we will include it in the transcript so people can email you directly about it. That would be fantastic. Are there any other studies at the James that you want to talk about? But I do want to also leave some time for some caller questions if people have questions.
I can quickly mention 2 trials since we are talking about multiply relapsed and refractory patients. There are 2 trials which include 2 monoclonal antibodies. One of these trials has this monoclonal antibody. Again, it's a phase 1 trial. I will mention that. It's an APRIL antibody. So the APRIL is a molecule which binds with the BCMA. BCMA is a receptor on the surface of plasma cells. So APRIL binds with the BCMA and increases the signaling through that BCMA protein. So this antibody targets the APRIL. So again, this is a phase 1 clinical trial, and we are enrolling here at the James in that trial. The second trial includes, again, monoclonal antibody. It's a very smart molecule. It's an antibody drug conjugate. So the antibody part targets CD38 on the surface of myeloma cells. So it's an anti-CD38 monoclonal antibody with a cytokine molecule attached to it, which is a toxic molecule. It's an interferon alpha. So this antibody delivers interferon alpha close to the myeloma cells by attaching itself to the myeloma. So both of these studies are phase 1 clinical trials for the patients who have relapsed on 3 or more lines of therapy.
Okay, and whose drug is that? I'm just curious.
So one drug, the antibody drug conjugate, is from Takeda. The other drug, the APRIL antibody, is from a pharmaceutical company called Bion.
Okay, and those are both phase 1 studies?
They are both phase 1 studies.
Well, it will be so interesting to see what happens, because hypothetically you could take 2 of these, like, this Takeda drug plus the GSK drug, and they're both these antibody drug conjugates that are delivering these toxic payloads, but to different targets. Of course, you have to test them, but that would be so fascinating.
Or target the BCMA with a BCMA antibody, as well as APRIL, which is the molecule that binds to the BCMA with an APRIL antibody. So 2 monoclonal antibodies. So I think these are great concepts, and that bring hope for the patients as well as for the clinicians.
Sure. It's really incredible to see the progress, and it's so exciting to watch this constant stream of new drugs be developed for multiple myeloma. It's not a club that any of us want to be in, but it is hopeful, and it's exciting that there's so much happening in myeloma.
That is true.
Well, that is wonderful. So we will have patients, if they're interested in these other 2, also email you for more information if they would like. And with that, we'd like to open it up for caller questions. So if you have a question for Dr. Chaudhry, you can call 347-637-2631 and press the number 1 on your keypad. And we have a caller question at 983-6757. So we'll start there. Go ahead with your question.
Hi, good morning, Dr. Chaudhry. This is Dana Holmes. How are you today? Thanks so much for taking the time. Appreciate it.
Thank you so much. I'm good.
Yes. I have a question concerning the BCMA expression. I understand that you do test for it, and it is reported, I guess, as being brightly expressed, and then they give you a percentage. And that's really testing the actual cells from the bone marrow biopsy. Is that correct?
Mm-hmm. That is true.
Okay. Is there any circulating BCMA within the serum that they test also?
So yes, you're right about that. So there is an enzyme that can break off that protein from the surface of plasma cells. So there is a circulating BCMA in the peripheral blood as well. And researchers have shown in past that high circulating BCMA is associated with poor prognosis in multiple myeloma. So in the trial, we did draw a lot of research blood and do a lot of testing and look at the serum as well as the bone marrow BCMA expression. So the expression has not panned out to be, to my knowledge, has not panned out to be associated with the degree of response as of now.
Okay, I see. And what about circulating plasma cells, the cells that actually manage to, I guess, become so aggressive that they leave the bone marrow microenvironment? And I guess that's how myeloma actually ends up metastasizing to other sites. How does a drug like this target those, or does it?
So that's a very good question. So plasma cells, when they break away, they lose a protein from their surface and then break away from the bone marrow microenvironment and come into the blood. When they exceed a certain percentage or certain number, it becomes a plasma cell leukemia. That's a patient who has a lot of plasma cells in the blood. So far, as the drug is experimental, these patients are not included in the trial.
Okay, I see. I understand. Is this the type of drug which you need to continue to use to keep myeloma under control? Does it have a half-life, or do you take it for a certain number of cycles and then you could stop it?
So unfortunately, as we consider myeloma incurable, and the patients on the trial have relapsed multiple times and are refractory to multiple therapy. So these patients stay on the drug as long as they can, depending on their tolerance as well as depending on the response. I will tell you that because of the eye toxicity, some patients had to have dose delays. So in the patients who had dose delays, the response continued. So if the dose was delayed, they didn't lose the benefit.
So you, because of the ocular toxicities, the drug needed to be discontinued for those side effects to actually resolve, but it didn't seem to have any effect on the efficacy of the previous. That's very interesting. Okay.
But then in most of the patients, they were able to restart the drug.
And did the side effects then come back, or was it just a transient which was corrected and then they were fine?
So the side effects were managed in different ways. So whenever the side effect goes above a certain level, we grade the side effects. Whenever it goes above a certain level, then the drug has to be, because it's an experimental drug, the drug has to be stopped, and the side effect has to be improved up to a certain level, and then the drug is restarted. And the side effects were managed with steroid eye drops. So in the phase 2 part, we are using the eye drops prophylactically, which means the patients will get the eye drops before they start the treatment.
That's a wonderful approach. Are the cells responsible for myeloma relapse being targeted by this type of drug? And do those cells always express BCMA, even perhaps at a lower level? I mean, do we even know which cell is responsible for relapse at this point?
So it's a very interesting question. So whenever myeloma relapses, it's very diverse and very genetically unstable. So again, the relapse is because of the plasma cells, essentially. And as a part of the screening studies, patients always get the bone marrow biopsy. And then we look at the BCMA expression in the bone marrow as well. And BCMA is universally expressed in all plasma cells, including cancerous plasma cells and other plasma cells as well.
Healthy plasma cells as well. Oh, I see. So this targets the healthy plasma cells as well.
It does.
So would that then present another line of side effects, so to speak? Because if you don't have healthy plasma cells, then you don't develop the antibodies to protect you from infection. So would a patient then require, let's say, IVIG treatment?
So that's a great question. We do IVIG treatment in patients who gets recurrent infections. But the plasma cells, if a drug targets the healthy plasma cells, those plasma cells can be replaced. So mature B cells can replace those plasma cells and still produce immunoglobulins.
Oh, they can. Okay.
Mm-hmm.
And do you actually see this drug moving into earlier disease trials? I mean, obviously, if it's effective in the relapsed and refractory, would you see it eventually? And if yes, how soon could we see it? Maybe even in newly diagnosed myeloma patients.
I think this drug has great potential, but we have to do the phase 2 trial as a single agent and then combine it in relapsed refractory patients before we move it to the front line.
Okay.
So I do see a future for this drug looking at the data that we have so far.
Very good. Dr. Chaudhry, thank you so very much for your time. And Jenny, thanks so much for permitting me to ask all these questions. Appreciate it very much. Have a great day, guys.
Great questions. Thanks. Okay, we have a caller at 209-9890. Go ahead with your question.
Yeah, thank you guys very much, Jenny, again, for all your work. This is Eric Hansen. I had a question on this BCMA target. There's been some research, I understand, that myeloma cells have the ability to either withdraw the BCMA target from the surface back into the cell so that it can't be, so it isn't expressed, or that it can actually shed this BCMA target from the surface so that it can evade these drugs that are targeting BCMA. Could you talk about that a little bit, Doctor?
That's a very interesting question. So the mechanism of resistance to this drug is still an area of ongoing research. So we still don't know when the patients stop responding. Why do they stop responding? I think some early research has shown that the patients, even after they become refractory to this drug, still have BCMA expression, which is a little bit different than other antigens like CD19 in lymphoma. So the plasma cells still express BCMA, but they develop other mechanisms to bypass that pathway and become resistant to the drug. But this still remains an area of ongoing research that why this happens. There is more information in terms of patients who have become refractory to CAR T cell, BCMA CAR T cell, but still remains an area of ongoing research.
Okay, thank you.
Mm-hmm.
Okay, thanks, Eric. That's a great question. Dr. Chaudhry, thank you so much for joining us today. We are just thrilled to learn more about this drug, and it sounds like a very interesting clinical trial that patients may want to consider, especially those who are relapsing on multiple lines of therapy. So we really appreciate you taking the time to talk to us about it in great detail, and it's just so nice being able to hear from you.
Thank you so much. It was a pleasure, Jenny. Thank you.
Well, we really, really appreciate your time and your focus on multiple myeloma and your care for all your patients. So we thank you.
Thank you. Okay.
And we thank our listeners for listening to Myeloma Crowd Radio. So we invite you to tune in next time to learn more about the latest in myeloma research and what it means for you.
With Lucky Land Slots, you can get lucky just about anywhere.
Dearly beloved, we are gathered here today to... Has anyone seen the bride and groom?
Sorry, we're here. We were getting lucky in the limo and we lost track of time.
No, Lucky Land Casino, with cash prizes that add up quicker than a guest registry.
In that case, I pronounce you lucky.
Play for free at LuckyLandSlots.com. Daily bonuses are waiting. No purchase necessary. Void where prohibited by law. 18+. Terms and conditions apply. See website for details.